Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, we are back with our questionnaire session where we are going to exercise C3 and C4 cycles, especially CAM cycle. We are going to look into photo respiration as well, that is C2 cycle. Excuse me. So yes, before entering into the questionnaire, I'd like to introduce the NEET World's special programs. Those are NEET World's 2 steps to medicine for 11th standard students with the minimal fee of 3333 per month with complete lecture sessions. Both offline and online mode are available and materials are also provided. And then NEET World's 1 step to medicine for 12th standard students who are aspiring for NEET 2026. For the minimal fee, again 3333, which is affordable for all the financial background students. And then Power Step to Medicine, this is for long term students, which is a unique program here with 3333 again with the most minimal fee. So all these programs are available both in the online and offline mode with all the study materials with the same fee. Now, how you can approach or get into these programs? You can get into these programs by dialing onto this number or directly visiting our branch, which is located over here in Narayanguda of Hyderabad. So this is all about the special programs of our NEET World. From when are they starting? Already one batch is running here and the next batch is going to begin or commence from July 1st, 2026. Okay. From July 1st, we are going to start one more new batch of the same minimal fee per month, which all these facilities provided. The unique thing about our institute is one-on-one mentorship is provided for all the students. So yes, students, to make use of it beta. This is the best program that I can say that is for all the students of Hyderabad. Clear, right, ma'am? Next. So yes, let's get started with the questions. First is statement based questions, just look into it. C4 plants will lack Calvin cycle. This is wrong because yesterday itself we have exercised that in C4 plants of bundle sheath cells, C4 plants which cells? Bundle sheath cells will conduct C3 cycle after decarboxylation of malic acid or ascorbic acid, that is 4 carbon compound, right. So after decarboxylation of this into 3 carbon compound and 1 carbon dioxide, this carbon dioxide will run the C3 cycle. Clear beta? So statement 1 is wrong. Look into this. C4 plants will lack photorespiration. This is correct. C4 plants will not have photorespiration. Photorespiration is also called as C2 cycle, which is a wasteful process. Photorespiration will operate only in the C3 plants. Photorespiration operates only in the C3 plants, not in the C4 plants. So C4 plants will lack photorespiration. That is correct statement. Next, C4 plants will have more productivity than C3 plants. True. C4 plants will have more productivity than C3 plants because in C4 plants there is no photorespiration and in C4 plants there is no photorespiration and there is an adaptation of double carbon dioxide fixation. 2 carboxylations will happen. 2 carboxylations will happen. One in mesophyll cells. First carboxylation happens in the mesophyll cells. That is first carboxylation, and the carbon dioxide acceptor in the mesophyll cells is what? PEP, and it will use the enzyme peptase, okay, by the enzyme peptase. Second carboxylation happens in where beta? Bundle sheath cells, and the acceptor of the second carboxylation in the second carboxylation is RUBP and the enzyme is Rubisco. Clear, ma? Fine, right? Now you see, ma. So C4 plants have more productivity than C3 plants. Yes, that is true. I have provided you the reasons why. Because of the absence of photorespiration or C2 cycle, and because of the two carboxylations that are happening, that is in the mesophyll cells and bundle sheath cells. And C4 plants are called as tropical plants, I have told you. C4 plants are what plants? Beta? Tropical plants. So they can tolerate the high temperatures, okay, they can tolerate high temperatures. So yes, this statement is true. Next, C4 plants cannot tolerate high temperatures, wrong. C4 plants can tolerate high temperatures, okay, because they are tropical plants. Now here in the question he is asking you how many of the above statements are incorrect, he is asking, ma'am. How many are incorrect? 1 is incorrect, 2 will be true only, 3 is correct and 4 is incorrect. So overall 2 statements are incorrect in this question, okay. First, look into all the reasons, all the explanations for each and every point that I have given you. This helps you to solve the further questions which we are doing now, okay. So here bundle sheath cells are present in where? Bundle sheath cells are cells which are present around the vascular bundles. If this is the vascular bundle, xylem and phloem, around this vascular bundles, bundle sheath cells are present. This bundle sheath cells are encoded by thick coating called as suberin, okay, they are encoded by thick coating called as suberin, and it is impervious for gaseous exchange, okay, it is impervious for gaseous exchange, and there are no intercellular spaces, okay. If you can see this bundle sheath cells, there are no intercellular spaces, no intercellular spaces. And then you might be having a doubt that if this is impervious to gaseous exchange, how come C4 acid, be it malic acid or ascorbic acid, is being entered from the mesophyll cells to bundle sheath cells? It is through plasmodesmatal connections, beta. It is through plasmodesmatal connections. Plasmodesmatal connections. Clear, ma? So here you can see vascular bundles are covered by bundle sheath cells in the form of wreath, okay, in the form of a wreath or garland. This arrangement or anatomy we will be calling as Kranz anatomy. This is called as Kranz anatomy. Fine, this is called as Kranz anatomy, and it is thickly coated by suberin, which is impervious for gaseous exchange, and there are no intercellular spaces. Most important points with respect to bundle sheath cells to be remembered. So here bundle sheath cells are present in C4 plants, and they are present around what? Vascular bundles in the form of a garland or a wreath. Clear, beta? So this is all about this question, and the right answer will be option A. Look into this question. Bundle sheath cells. Bundle sheath cells will allow gaseous exchange, have intercellular spaces, wrong, they do not have intercellular spaces. They have large number of chloroplasts, true. They do not allow gaseous exchange because they have thick suberin coatings over it. So these two statements are wrong, C is true. And what kind of chloroplasts you will see in the bundle sheath cells, beta? What kind of chloroplasts? Agranal chloroplasts, okay, you call it as agranal chloroplast. Fine, agranal chloroplast, there is no grana inside the chloroplast, complete stroma only will be present, and there are stroma lamellae present inside this bundle sheath cells chloroplast, okay, stroma lamellae. As a stroma lamellae is present, what photosystem can be present in the bundle sheath cells, beta? PS1, photosystem 1 is present. So yes, it has large number of chloroplasts, those are agranal chloroplasts, grana is completely absent, but it has stroma lamellae, it has PS1. Clear, right, ma? Next question. Which of the following is true? C4 plants will lack rubisco. Wrong. C4 plants will have rubisco in which cells? Online students, you can answer me. Okay, in which cells C4 plants will be having rubisco? It is present in the bundle sheath cells. Mesophyll cells of C4 plants will lack rubisco. Okay, mesophyll cells of C4 plants, it will not be having rubisco. That is correct. Okay, because it is having what? Pepcase. Okay, it doesn't have rubisco, it has pepcase. Mesophyll cells of C4 plants will have pepcase, it doesn't have rubisco. So it lacks rubisco, that statement is right now. Next, bundle sheath cells of C4 plants will lack rubisco. This is wrong because bundle sheath cells will be having rubisco. C3 plants will lack rubisco. Wrong. C3 plants will be having rubisco. So here what will be the right answer beta? Mesophyll cells of C4 plants will lack rubisco. That will be the right answer. Next question. Oxaloacetic acid forms other 4 carbon acids which are transported. What are they? See, in mesophyll cells, once after the carboxylation happens, that carbon dioxide which is accepted by phosphoenol pyruvate, which is a 3 carbon compound, and the enzyme that is catalyzing is pepcase. Pepcase is the enzyme which is catalyzing this, and this forms oxaloacetic acid. This forms oxaloacetic acid which is a 4 carbon compound. Okay, this will be converted to other organic acids which are isomerized, we can say. These are isomerized to other carbonic acids which are 4 carbon only. They are like malic acid and ascorbic acid. Malic acid or ascorbic acids. Okay, this malic acid or ascorbic acid, through plasmodesmatal connections, they will enter into bundle sheath cells, and this C4 carbon compounds, organic acids, will get decarboxylated into C3 and carbon dioxide. This will be regenerated back to PEP. This is regenerated back to PEP, and this carbon dioxide will run what cycle? Kelvin cycle. Clear beta, right? Understood na? So here what acids it is getting converted to what carbonic acids? Malic acid and aspartic acid. Clear, right? Next, this is aspartic acid, sorry, malic acid and aspartic acid. Next, Kelvin cycle takes place in dash and in all C3 plants and in all C4 plants. Kelvin cycle takes place in the mesophyll cells of C3 plants. Mesophyll cells of C3 plants, and what cells of C4 plants beta? Bundle sheath cells of C4 plants. It is as simple as it is, right? So it is Kelvin cycle takes place in mesophyll cells of C3 plants and bundle sheath cells of C4 plants. So this will be the correct answer. Clear, right, ma'am? Next. So now whatever questions we are going to exercise are purely based on photorespiration. So I would like to give you complete gist on photorespiration once you listen. Photorespiration is also called as C2 cycle, and this is operated only in C3 plants. This is operated or seen only in the C3 plants. Okay, this is seen only in the C3 plants. So here in this photorespiration, what happens is, first in the chloroplast, when first of all you have to understand the first step, primary step of C3 cycle, that is whenever carbon dioxide is being entered to the cell, this carbon dioxide is primarily accepted by 5 carbon compound RuBP, we all know that. And what is the enzyme that is catalyzing? Rubisco is the enzyme. What is the enzyme beta? Rubisco. This Rubisco, what is the full name of this Rubisco? Ribulose bisphosphate carboxylase oxygenase. This has more affinity to bind to carbon dioxide than that of oxygen. More affinity or more equally affinity will be present. So Rubisco has high affinity to bind with carbon dioxide and more equal affinity likely with carbon dioxide. After that oxygen also it will bind. Whenever there are low carbon dioxide concentrations, what is the optimum carbon dioxide concentration in factors affecting photosynthesis? We learn that optimum carbon dioxide concentration for photosynthesis will be C3 that is 0.03 microliters per liter. Okay, that is 0.03 of carbon dioxide is optimal, right? Less than that, if carbon dioxide is present, then Rubisco will bind with oxygen. C2 cycle is operated when Rubisco is binding with oxygen despite of carbon dioxide. Fine. So, Rubisco is operated only in the C3 plants, C2 cycle is operated only in the C3 plants because Rubisco will bind both with oxygen and carbon dioxide. When it comes to the affinity, it has higher affinity for carbon dioxide than oxygen. Clear? Whenever carbon dioxide concentrations are lower than the optimum like 0.03, then it will bind with oxygen. Okay, Rubisco will bind with oxygen. Once Rubisco will bind with oxygen, then what happens is when Rubisco is binding with oxygen, then two molecules will be formed. Okay, two molecules will be formed when RUBP along with oxygen, Rubisco binds that and it forms one 3-carbon compound, that is phosphoglyceric acid, PGA. Okay, that is phosphoglyceric acid, and one 2-carbon compound, that is phosphoglycolate. So, if there are two molecules of phosphoglyceric acid, it will turn on to the Kelvin cycle, that is reduction. But then here, as there is only one phosphoglyceric acid, to get one more phosphoglyceric acid, phosphoglycolate has to travel across 3 organelles, and it consumes so much of energy. It will not release the energy, it will not release the sucrose, rather it consumes oxygen and releases of carbon dioxide. That is why C2 cycle or photorespiration is said to be a wasteful process. I will explain you in detail, just have a look at it. This phosphoglycolate will turn to glycolate. Glycolate will enter into the next organelle, that is peroxisome. Okay, it will enter into the next organelle, that is peroxisome. Glycolate along with hydrogen peroxide, it converts to glycine. Glycine enters to the next organelle, that is mitochondria. Utilizing NAD+, it converts to NADH2 and glycine gets converted to serine over here. Here decarboxylation happens, ammonia will be released as well. Here if you see, serine again enters to the peroxisomes, converts to hydroxypyruvic acid, then glyceric acid again enters to chloroplast. By consuming the ATP, by utilizing ATP, it phosphorylates the glyceric acid and gives rise to phosphoglyceric acid. So, to produce one more phosphoglyceric acid, this glycolate has to travel across peroxisome, mitochondria, again peroxisome, and then it lands the chloroplast. In this process, carbon dioxide is evolved, energy is being utilized, no sugar molecule is formed, rather oxygen is being utilized and carbon dioxide is being evolved. That is the reason C2 cycle is a wasteful process that is only operated in the C3 plants. This is all about C2 cycle beta. Any doubts anywhere, you can message in the chat box. Now we are going to exercise questions on the photorespiration itself. Look at it. This is a statement based question. Read the following. Okay, read the following statements. Ribulose bisphosphate is most abundant enzyme in the world. Wrong. Ribulose bisphosphate carboxylase oxygenase is the most abundant enzyme in this world. Okay, don't get confused between RUBP and Rubisco. This is the world's abundant enzyme and this is the primary acceptor of carbon dioxide in C3 plants. Remember that beta. Don't get confused between RUBP and Rubisco. This is ribulose bisphosphate, which is the primary acceptor of carbon dioxide in C3 plants. This is Rubisco, which is ribulose bisphosphate carboxylase oxygenase. Fine, which is the world's abundant enzyme. So this is a wrong statement. Statement B you look into, photorespiration doesn't occur in C4 plants. That is 100% correct. Okay. So here you will go with the option A is incorrect and B is correct. Option B will be the right answer. Next question you see beta. Rubisco has dash when carbon dioxide and oxygen are equal. I already told you Rubisco when carbon dioxide and oxygen are in equal concentration, as Rubisco has higher affinity to bind with carbon dioxide, it will bind with carbon dioxide itself. It has greater affinity for carbon dioxide than for oxygen. So A will be correct. Okay. And Rubisco, if at all carbon dioxide is less and oxygen is more, then Rubisco will bind with oxygen itself. When both are equal, it binds with carbon dioxide only, okay, because it has higher affinity for carbon dioxide. Look into this question, ma. In C3 plants, some oxygen binds to Rubisco and carbon dioxide fixation will increase. Carbon dioxide fixation will decrease because when oxygen binds to Rubisco, carbon dioxide fixation will not happen easily because it has to travel through 3 organelles and that big cycle will be operated. So, carbon dioxide concentration will be decreased, not increases. No oxygen binds to Rubisco. True, there is no oxygen, that is no oxygen binds to Rubisco. Wrong. Oxygen can bind to Rubisco. This you see third statement. Some oxygen binds to Rubisco and carbon dioxide fixation decreases. True. When oxygen binds to Rubisco, carbon dioxide fixation will decrease or carboxylation will decrease. Only oxygen binds to Rubisco. Wrong. Both oxygen and carbon dioxide will bind to Rubisco. Fine. So, option C will be correct over here. Next question. When oxygen binds to Rubisco, RUBP is converted to. Remember the first step of photorespiration beta. When oxygen is bind to RUBP, what happens? Rubisco is binding with oxygen. As a result, you will get one molecule of 3 carbon compound, that is phosphoglyceric acid, and one molecule of 2 carbon compound, that is phosphoglycolate. Clear ma, right? So, here it produces one molecule of 3 carbon compound, that is phosphoglycerate, true, and one is phosphoglycolate. Okay, the phosphoglycerate and phosphoenol pyruvate, wrong. Yeah, here you see phosphoglycolate and phosphoglycerate. This is 2 carbon and this is 3 carbon. Clear now? As 2 carbon compound is formed as a primary product, this is also called as C2 cycle. That's it. This you see in photorespiration, sugar and ATP are formed? No. No ATP is formed, no sugar is formed, no oxygen is evolved. Rather, ATP is utilized, carbon dioxide is evolved. Okay, that is the reason it is a wasteful process, right? So, remember that and answer this question, ma. Any one of you from online can answer. In photorespiration, sugar and ATP are formed. Wrong. Sugar is formed but not ATP, wrong. ATP but not sugar. Sugar and ATP are not formed. This will be the correct answer. Here you see assertion and reason type of question. Photorespiration doesn't occur in C4 plants. True, because it has an adaptation of bundle sheath cells and the primary acceptor is pepcase. It doesn't have any affinity to bind with oxygen. Okay. So, that is why that is an adaptation. So, here in C4 plants there is no possibility of photorespiration. In reason what it is provided, carbon dioxide concentration at enzyme site is higher in C4 plants. True. Okay. Carbon dioxide concentration at the enzyme site, that is pepcase, is correct. But reason should not be this, right? What should be the reason? Photorespiration doesn't occur in C4 plants, that is because pepcase doesn't have any affinity for oxygen. The statement is true, but it is not correctly explaining the reason. In that case, what option we have to choose, beta? Both assertion and reason are true and it is not the correct explanation of assertion. Might be it is not given, you take as option A, it is not correct explanation of assertion. In explanation, what should be there, beta? Pepcase has no affinity to bind with oxygen. That is the reason it is not operated in C4 plants. This you see. Next question. Glycolate accumulates in chloroplast when there is. Glycolate accumulates in chloroplast. He is talking about which cycle? C2 cycle only. That is first step. Glycolate accumulates when there is high carbon dioxide? No. When there is high carbon dioxide, why Rubisco will bind to oxygen? No. Bright light? No. Low temperature? No. When there is low carbon dioxide, then only Rubisco will bind with oxygen. Then glycolate will accumulate in chloroplast. So, it is option D. Next, in CAM plants, the concentration of organic acids. What will be the concentration of organic acids in the CAM plants? At what time it will be more? Once we will recall the CAM cycle, beta, we know CAM cycle in CAMs to activate the CAM cycle or CAM cycle is activated in CAM plants. Crassulacean acid metabolism is activated over here, and what are CAM plants? Nothing but the desert plants. Okay, these are desert plants like Bryophyllum, Colonchi, okay, cactus, Opuntia, these are the best examples. So, here in desert plants what is present? Scotoactive stomata, the stomata which opens during the night time. The stomata which opens during night time where carbon dioxide will enter, and here the primary acceptor is also PEP by the enzyme pepcase. This is being fixed to oxaloacetic acid, and this gets converted to organic acids like malic acid or aspartic acid. Okay, malic acid or aspartic acid. This conversion into malic acid or aspartic acid takes place during night time only. Okay, and these are stored inside the vacuoles. Okay, inside the vacuole. And during daytime C3 cycle is operated. During night time C4 cycle is operated in CAM plants. So, here you see ma, in CAM plants the concentration of organic acids will increase at night. This will increase during night time, not during the day. Fine. Next you see. This is the last question that we are going to do today. So, look into this question ma, assertion reason type again. Photorespiration is completed in the 3 cell organelles. Tell me what are those 3 cell organelles? First is chloroplast, second is peroxisome, third will be glyoxysomes. Glyoxysomes. Clear? So, you have chloroplast, peroxysomes and glyoxysomes. This is correct itself. In reason, peroxysomes, glyoxysomes and desmosomes are involved in photorespiration. Reason is completely wrong because chloroplast, peroxysomes and mitochondria is involved. I am sorry. Chloroplast, peroxysomes and mitochondria. Because in mitochondria only glycine is converted to serine and it utilizes NAD plus to the release of NADH, And decarboxylation, release of ammonia, everything happens in mitochondria itself, right? So sorry, it is not glyoxysomes, it is mitochondria. So organelles are wrong, right? Peroxisomes, chloroplast and mitochondria. It is not glyoxysomes and desmides. So here assertion is true and reason is false. So we have to go with option C. Clear, ma? So yes, I would like to end the session by again giving you a brief account on the courses that we are offering, the special courses that we are offering in the NEET World. Those are NEET World's 2 Steps to Medicine for 11th standard students who are targeted for NEET 2027, which is a 2-year program with the minimal fee of 3333, and NEET World's 1 Step to Medicine, which is for 12th standard students who target NEET 2026 with the same fee of 3333, and for long term students you land on the program that is Power Step to Medicine with the fee 3333 per month again. So complete course both offline and online mode with the same fee, and materials are provided as well. Clear? So whoever are interested can call on to the given number, or you can directly visit our branch at this address in Narayanguda, Hyderabad. Clear, ma? And this I have already told you that new batch is going to start from July 1st. Fine. Finally, I would like to say you, all the students in the public platform, you will get highly benefited if you join the Telegram group. Just by scanning this QR code, you will be added into NEET World's Telegram channel, where you will get daily quizzes for botany, zoology, physics and chemistry, for maths also to the students who want to write JEE mains, and then live class schedules in the YouTube will be updated in the Telegram channel, and free resources will be provided, free materials, free PDFs, free doubt solving sessions, everything are provided. One-on-one doubt solving sessions with faculties will be provided if you just call on to this number given below. So NEET World will be your ultimate destination for NEET, JEE, MCET or CUT exams, any of this. Just log into the Telegram channel and call on to this number. You are freely advised, okay? Any sort of advice you need regarding these programs or coaching, anything, we are here to solve it, okay? So thank you, meet you tomorrow.